Hello, everyone, and welcome to today's webinar, Part 2, Sigma Strong, How Stronger Harmonization Builds Better Assays, Simplifies Quality Control, and Strengthens Results. I am Michelle Ashton of Laboratories, and I'll be your moderator for today's event. Today's educational web seminar is presented by Laboratories and brought to you by Abbott. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker. Sten Westgard is the Director of Client Services and Technology for Westgard Quality Control. Mr. Westgard earned his master's degree in computer science from Pace University, New York. For nearly 25 years, Sten has managed the Westgard website, course portal, and blog, creating and administering online training, as well as editing and writing hundreds of reports, essays, and applications on quality control, method validation, Six Sigma risk management, and other laboratory management topics. Sten Westgard has edited and contributed to numerous books on quality, QC practices, method validation, Six Sigma QC design and control, Six Sigma risk analysis, and CLIA final rules, and co-edited special issues of Clinics in Laboratory Medicine and Biochimica Medica. Sten is also an adjunct faculty member of the Mayo Clinic School of Health Sciences, adjunct faculty member of the University of Alexandria, Egypt, and adjunct visiting faculty member of University in India, and an honorary visiting professor at a university in Shanghai. Sten Westgard will now begin his presentation.
Welcome. This episode or this session is going to be about Six Sigma and about getting your Sigma to be not only strong, but stronger. I want to start by making a disclosure. I'm thankful for the opportunity to connect with you today. Abbott is providing the support for not only my time, but for the whole connection between you and me and all of the audience. The focus of today is on stronger harmonization and how it can build better assays and simplify your QC and strengthen the results you deliver to patients. We want to start by understanding what Six Sigma is and why it's not a simple destination. You don't arrive at it and then you're finished. It's really a pathway. It's a journey, and you must sustain Six Sigma, not just one day, but every day. We'll teach you how to calculate an analytical Six Sigma metric. We'll help you identify improvements in assay performance through the lens of Six Sigma metrics. And then we'll use those Six Sigma metrics to design QC in a better, more efficient way. And the tool that will provide that to you will be the Westgard Sigma rules, the latest evolution of Westgard rules. Now there's also a rather more important disclosure, since many of you might have tuned into this session not knowing which Westgard was going to be delivering the lecture. I do want to clarify that for you. You are listening to a Westgard, that is Sten Westgard. I'm only about 25 years into my career. So I've only been doing this for a quarter of a century. The fellow who's done it for more than 40 years at the University of Wisconsin, and really more than 50 years of his entire career, is D. Westgard, James O. Westgard, the real father of the Westgard rules, and myself. Together we have put a resource out to the public at westgard.com and westgard.org. So when we're not giving presentations, we're trying to make as much material available to laboratory professionals around the world. Our flagship at westgard.com has more than 70,000 professional members, and there are more than 600 articles available for free. It's a free membership, and you can download things, you can have spreadsheets, graphing tools, anything on the Westgard rules, many, many examples of Six Sigma. We have training, we have social media as well. And in this particular moment, I think no one can deny that quality has become paramount. Even if we look at the tools we've been using to fight the global pandemic, just a little over a month ago, the number of methods on the market topped more than 1,100. But of those, only a few hundred are actually allowed in the United States by the FDA. And if we focus on the serology methods, the antibody tests, only 79 were approved as of May, whereas 264 antibody methods were on a do not distribute list. That is, 3 out of 4 methods are rejected by the FDA for use in fighting this pandemic. A majority of methods on the market are not good enough. That's a frightening reality, because if those methods are in laboratories around the world, well, then we might be failing at quality, we might be breaking the trust with our clinicians and patients, and really it has severe consequences to the public health. But let's take a step back, not look at 2020 and 2021, but let's go back many more decades. Some of you may remember these things: pipetting by mouth, or the use of flame photometers, or the SMAC, the first automated instrument from Technicon. And then there is the use of 2 SD control limits. All of these things are decades old. Some of them are half a century old. But probably one of those techniques seems like it's not old, it's something you're doing right now. But really, if you don't pipette by mouth anymore, you also shouldn't be using 2 SD control limits. When we look at a modern survey of QC practices, we find there are 3 main pains or bad habits. The use of limits that are too tight, known as anorexic QC, the repeating of controls over and over, known as gambler QC, or the use of ranges or limits that are far wider than what an individual laboratory should have, and we call that blind man QC. In our surveys, not only in 2017, but even our ongoing survey this year, we find that every laboratory, I should say every country in the world, has these problems. Every type of laboratory has these problems. And all of these bad habits burn up time and money and cause a lot of unnecessary frustration. For instance, if we look at the use of 2 SD practices, even though it's really a half-century old tradition, not science, a majority of labs are using 2 SD limits. And the problem with 2 SD limits, it's not just that it's been done for 50 years, it's that the false rejection rates from 2SD limits are incredibly high. If you're using 3 controls, it's a 14% false rejection rate. If you're using 2 controls, it's about 1 in 10 runs are expected to be out of control, even when nothing is wrong with the method. So that means even when the method is operating perfectly, we're placing an unnecessary burden on our shoulders. We're saying, yeah, the method could be perfect, but 1 out of 10 times we need you to chase down an out-of-control event that is essentially a ghost. That might have been a practice that was marginally acceptable back 50 years ago, but in today's modern laboratory, 1 out of 10 runs cannot be out of control. That's just too much waste. So we need to change QC, and that's one of the reasons why the Westgard rules came around in 1981, which was 40 years ago. And the Westgard rules reduced the false rejection from the 1-2S approach. It maximized error detection from just a few measurements, and it also helped figure out when something was wrong, what might be wrong, whether it could troubleshoot or triage the kind of out-of-control event into either a random or a systematic error. The benefits from Westgard rules were immediately felt because instead of 9% or 14% false rejection rates, they were lowered to 3% and 1%. The adoption and implementation of Westgard rules really dropped the number of outliers and dropped the number of troubleshooting events that labs had to go through. And that's why the adoption was so fast and so global and so quick. But those rules from 40 years ago are not the rules we recommend today. Over the decades, we have changed how the Westgard rules are recommended to people. For instance, we no longer recommend using the 1-2S warning rule. Again, because even if you only treat a 1-2S outlier as a warning, that warning goes off so many times, you might grow deaf to it. You might stop listening and ignoring it. So instead, we now recommend a modern set with no 1-2S warning, but instead you go straight to the rejection rules. And not only do we have those optimized for 2 controls, but also for 3 controls. For situations with some assays, particularly in hematology, when you might be running 3 levels of control, use a 2 out of 3 2S, or a 3 1S, or a 6X. And then even more recently, we've adopted 6 Sigma into the Westgard rules themselves. So 6 Sigma, Westgard rules, you put them together, you have Westgard Sigma rules. And what you'll notice is the rules look pretty familiar. It's the bottom of the graphic that has changed. You now see little dotted lines and delineations for 6 Sigma, 5 Sigma, 4 Sigma, and 3 Sigma. And essentially what you need to know is the sigma of your test, and if you know that, you will know how many Westgard rules you need to use. And what you might be able to see here is that at 6 Sigma, you don't need all the Westgard rules. You just need 1 rule. It's not a multi-rule approach, it's a single rule approach. And you can see at the other side, 3 Sigma is when you need all the Westgard rules. So this is one way we can reduce the burden of QC on laboratories. As laboratories have better and better quality in their assays, they do fewer and fewer Westgard rules. But as we've introduced the Westgard Sigma rules, we probably need to cover the concept of 6 Sigma. 6 Sigma is really a technique that allows us to prevent our use of bad methods. So we won't adopt or bring into our laboratory a bad quality method, and we will also not do any of these bad QC habits. Six Sigma is the goal of the approach. And essentially it means out of 1 million reportable results, you have less than 4 defects. And Six Sigma has been used many places, many different industries, and the achievement of Six Sigma is found to be associated with the best efficiency, the most satisfied customers, the greatest reliability, and the highest profitability. If it helps to understand what kind of processes are Six Sigma, think about airline safety. I don't know if you remember when we all used to go onto planes or not. Maybe we will be able to do this again soon. But in the US at least, there are less than 3.4 crashes per million departures. So safety is actually better than Six Sigma in the US, or it was before the pandemic. Two things in the laboratory: the number of samples that get damaged in transport, or the number of samples that aren't stored properly before analysis. In an IFCC assessment of pre-analytical quality, those two processes came in at 6 sigma. 5 sigma is considered excellent. It's really not that many defects. It's 233 now per million reportables. And this is closer to how often we see a misidentified test request or how often samples are misidentified. 4 sigma is considered good performance. Now we're at 6,000 defects per million. And that's closer to the rate of misplaced checked luggage. So how often does your checked bag take a different vacation than you do? About 1 in 300. How often does your specimen come in and not have enough sample volume to actually run all the tests that have been ordered? How often is your sample hemolyzed? Well, we know that's a very chronic problem for many laboratories, and that is about a 4 sigma process. 3 sigma is the minimum acceptable performance. Now we're at 67,000 defects per million. And just to, again, associate it with something that happens in the laboratory, how often are your microbiology samples contaminated? Again, that's a chronic problem, and it really is the minimum acceptable quality in most laboratories. Below 3 sigma is considered unacceptable. Now we're in the hundreds of thousands of defects. If we get below 1 sigma, we're worse than a coin flip. Departure delays for the U.S. airlines, which are around 30% or were around 30%, that's a below 3 sigma process. And that helps us understand why we are often frustrated with airlines and hate the way they treat us, and yet we feel extremely safe about getting on a flight, and we're very confident that we'll be able to get off that plane at the end of the trip. In the laboratory, when we want to measure 6 sigma performance, we need to know imprecision and inaccuracy, your CV and your bias. These are gained from your controls and either through your EQA or proficiency testing or your peer group. And then you can put those together along with the total allowable error, which you can look up many different places where that's located, but the Westgard.com website has most of those resources available in a free section. You take the allowable total error, you subtract the absolute value of your bias, and you divide it by the CV. And with that, you will now have a sigma metric. Again, once you know the sigma metric, then you can come to the Westgard sigma rules, and if it's a 5 sigma, you look where that dotted line goes, and it's now you know that you need 3 Westgard rules: the 1,3s, 2,2s, R4s. And if you look closely at this particular graph, you will notice that there not only are the 6 sigma delineations, but there's also suggested run sizes. just in the last few years, we've been able to adapt the Westgard sigma rules to now give you QC frequency. The mathematics to determine your optimal QC frequency now exist, and we've incorporated them into the Westgard sigma rules. Another way to look at the sigma performance is to use what we call a method decision chart. And this is just a visual way to understand sigma metrics, taking the imprecision along the x-axis and the bias along the y-axis. And the diagonal lines break up the graph into different sigma zones, with 6 sigma being closer to the graph's origin. That's what's labeled world-class. 5 sigma is excellent, 4 sigma is good, 3 sigma is marginal. Remember, it's the minimum acceptable quality. 2 sigma is poor, and less than 2 sigma, I hope we can all agree, is unacceptable. It's easier to think about it as the targets, just like we were looking at earlier. The center of the bullseye is the 6 sigma, and the closer you get to it, the fewer defects you have, and the more confidence you and your clinician and your patient can have with the veracity of the results. But as you have more imprecision, as you have more bias, that means more defects are being generated, more noise is starting to contaminate all of the results, and near the graph, near the upper right quadrant of this graph, really you're reaching a point where you're confusing clinicians more than helping to confirm a diagnosis. And this graph can be found in a free download as well as an online tool for free at the Westgard website. Now let's talk about standardization and harmonization. We always talk about we don't want to be comparing apples to oranges. There's a much more scientific way to talk about that, not just apples and oranges, but really metrology. And many of you may have seen this. This is the ladder, or sometimes I call it the chutes and ladder of metrology and standardization. At the top is the pure, true measurement, and then there's all these different steps that get taken as you move from standard or truth down to the manufacturing of the method and the calibration of the method, and then the routine result down at the bottom. And the more steps you take, the more possible you get further away from the truth. There's a lot of terminology associated with this. There's standardization, there's harmonization, which is what you do when you have no standard, but you still want results to be close to each other. There's traceability, which is how good are you at being closer and closer to that true answer. And then uncertainty, which is as you get further down, if you go down those chutes, you get more and more uncertainty. And obviously you want as little uncertainty as possible in that result. It's not possible to avoid all uncertainty, but you want to minimize it as much as possible. The benefits from having a standardized or harmonized assay really accrue to the patients first and foremost. If you have results that are stable across labs and platforms and instruments and methods, then the patient can move through a health system and not have to worry that they were tested on one instrument at this hospital and then another instrument at another hospital, and those two results are incomparable. Therefore, we can't understand what's going on with the patient without baselining them first. So this will be much better if we could be standardized and harmonized for the patients. It would also be really good for laboratories, because then it becomes easier to interchange the methods and instruments. It also means not only is there a standard true answer that you're delivering, but you have a standard level of quality that you can rely on. Manufacturers, strangely, or I suppose on purpose, they have the least amount to gain from standardization and harmonization. They do it because it's good for the patient. They do it because it's good for their labs, for the customers. But really, the more we do this, the more we commodify the assay, and that will mean it's the quality of the assay becomes less important because we've all achieved the requisite level of quality. So I do view this as a victory in that science trumps commercial interests here. The pursuit of standardization and harmonization is a pure good for patients. Manufacturers, what they probably would like most of all is, here's a method and no one else can do it. It's only us, and therefore they have a monopoly on the market. But luckily, through our professionalism, through our science, we push all the manufacturers to make their methods better and closer to each other. It's good for us in the lab, and it's great for the patients. There is an underlying assumption that each new wave of standardization and harmonization will improve the analytical quality. And for the most part, that is observed and seen, yes, as assays get better, as assays get closer to a standard, they are getting better in quality. It is, however, possible, and if we look at a lot of Chinese products flooding the market, we may accidentally get standardized to a bad level of quality. The standardization approach is let's get better to truth and better to high quality. Now here's where our method decision charts come into play. And what I'm showing you is 6 different assays, and we're going to see them in the next slide as well. This is current performance on an alinity at Winchester Medical Center in the US. So this is the current level of quality for these assays, except urea is not plotted because urea was very poor in performance. And in our sigma verification program, if you're below 4 sigma, we don't plot you. We won't qualify them for our program. Here are the new sigma strong assays, albumin, BCG, and BCP, and amylase, and cholesterol, and creatinine, and total protein, and urea, and uric acid. We can see this new wave of assays. Almost all of them are at 6 sigma. And then one is at 5 and one is at 4. One of the interesting things, not only are these assays being standardized to better and better truth, but they're also being given tighter and tighter goals for quality. So in the previous set of assays, amylase was only being held to a 30% allowable total error, which is the CLIA standard. But a RICO standard is now being imposed, which is essentially having the quality requirement, and it's 15.7% now. The creatinine goal could have been 35.4% by CLIA at that particular level where we were measuring, but actually they're imposing a 20% goal. This is from the National Kidney Disease Education Program. And the 17% goal, which is standard by CLIA, has been replaced by a 13% goal. This is pretty unusual to see a manufacturer, not only do they re-standardize the assay, moving it closer to truth, but then they tighten the goal. They say, we're not going to try and hit the old target, we're going to try and hit a new and more difficult target. That's really impressive, and just to be honest, I have rarely seen a manufacturer be this hard on themselves when developing a new assay. So real credit to these Sigma Strong assays for adopting tighter goals. Here's an example of the difference. So on the albumin, the amylase assay, a 1992 CLIA goal has been replaced by a RICOS goal. And you can see it doesn't matter that you've shrunk the goal, you're still in the bullseye. It's still at Six Sigma quality. For creatinine, the CLIA goal of 1992 was first used, and then the tighter goal, which is by the National Kidney Disease Education Program, was imposed. So the impact of that means it's not a world-class assay, it's now an excellent assay. The uric acid, that assay, when benchmarked against the original CLIA goal, is Six Sigma, but under the tightened goal, which is now really Bach, which is the German, a German set of allowable total errors, even when a tighter goal from the Germans are imposed, it is still a Six Sigma assay. Now, we're not trying to hit smaller and smaller goals just for the ability to say we're hitting these goals. There is a payoff to a better assay. And if you were using Westgard rules in the classic formulation, so you have the warning rule, you're running two controls, and with the 8x, you're looking across 4 runs. If you actually benchmark the quality of your assay and use Westgard Sigma rules to redesign how you do QC, well, if you happen to achieve Six Sigma, and as we saw in the previous slides, almost all these assays are at Six Sigma, then you can reduce the number of rules you implement by more than 80%. And you can reduce the number of runs of QC by 75%. And how about that false rejection? Instead of it being 9% or 14% from 2SD, now you're almost going to completely eliminate the false rejection. I know that sounds almost fanciful, that you could reduce all of your QC effort by that much, but we have a decade of experience in the field where it is being done. So here's Winchester Medical Center at Valley Health. This is in Virginia, and for the 9-year lifetime of their Architect instruments, when they implemented this approach, they found an 85% reduction in their QC failures. And they found that there was an hour less a day that they had to spend working that instrument. And when you quantify all of those hours into a savings, the labor savings is more than $100,000 over the lifetime of the instruments. And that's certainly something that makes the laboratory happy, the idea that you don't have to be standing in front of the box all those hours and repeating controls and running new controls and recalibrating and calling technical support. All of those things go away. But also turned out that there was a hard dollar savings because they ordered fewer controls and they consumed fewer supplies and they burned less reagent by running fewer, because they had fewer controls to run. And over the lifespan of the instruments, 9 years, that was 160,000 hard dollar savings. So you put those soft and hard together, that's $250,000 you save over the long run on these instruments. And this is something you can actually demonstrate to your administration or your manager. High quality does actually save you money, and it does not, this kind of money savings does not sacrifice the quality of the result. So in conclusion, we do see, like everything in the laboratory, there's always constant change. And it will happen with the Westgard rules too. And it will happen with how you run your QC. But those changes are going to be for the better. The results you will know just how reliable they are. You can back up, if a clinician questions a particular result, you can tell them, look, this is a Six Sigma assay. I only expect to be wrong 4 times out of a million. And then the QC can get much easier for you. You'll have fewer rules. You'll run fewer controls. You may even be able to run less often. The Westgard Sigma rules are a way to simply look at all of these savings and benefits you get out of Six Sigma. And again, through the new wave of assays that are better standardized and better in performance, that will increase the benefits of using Six Sigma, of using Westgard Sigma rules, will reduce even further the number of out-of-control events you have, will reduce even further the amount of supplies and reagents you need to consume, and reduce even further the amount of time you have to spend chasing out-of-control events. And with that, I want to thank you for your time. Thanks for joining this session, and I know we're going to have a question and answer session next. Thanks again to Abbott and Laboratories for supporting us and enabling us to be able to connect to all of you. Finally, you'll see that email address down there, sten@westgard.com. Part of the mission of Westgard is not just to present the tools, but also to help labs implement them. So I encourage you to ask questions in the Q&A session to follow, but if something occurs to you later as you're digesting and ruminating on these lessons, please feel free to reach out and email me. Thank you.
All right, thank you, Sten, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's go ahead and get started. Your first question, Sten, what is the best way for labs to determine precision to calculate the sigma value? Must I do multiple days and multiple lots to get a value?
That's a great question. Thank you very much, Michelle. Glad to see so many questions coming in. For precision, in our own program of sigma verification, we ask for 100 data points, and that could cover 3 months on a chemistry if you're running once a day. If you're running more often, then that's a shorter time period. And that's kind of in line with the CLSI C24 document, which is that expresses a lot of the best practices for QC, and it recommends getting an estimate of 3 to 6 months of data to give yourself a cumulative CV and a cumulative mean. So we're matching that at least on the minimum side. Do you have to do multiple reagent lots? Well, that could be nice, and certainly if you're doing this on the hematology or other tests where you have very short reagent lot life, probably that's just going to happen naturally as you try and get so many data points. But on other instruments, I would say with a lot of chemistries where you have very long shelf life, even for the reagents, that you may not have to get multiple reagent lots. If it's a very stable assay, you probably don't need that extra information.
Okay.
Go ahead for the next question.
Wonderful. All right. Where is this approach usable?
That is a very broad question, so let's first talk about where in the world it could be used. And in our, again, our Sigma verification program, we have labs all over. So we have them in Asia, we have them in India, we have them in Europe, we have them in South America, we have them in Russia, we have them in North America. So Sigma metrics formally are being used almost everywhere, but maybe that's on a country or region basis. Within a single laboratory, we see Sigma metrics being used most often in the chemistry. Immunoassays is probably the second most popular area, and then from that point on, toxicology, endocrinology, and we're starting to do work to try and move it into the serology testing area to make adaptations for semi-quantitative assays. Now, to step back again, the sigma equation is agnostic. So if you say, I have an allowable total error, I have a bias, I have a CV, the sigma equation doesn't say, well, wait a minute, where are you from? And then start rejecting you, oh no, you're from the wrong country, or you're the wrong kind of test. The equation itself, if you give it numbers that make sense, that are realistic, it will give you a sigma metric. So the application of this approach can be quite broad.
All right, thank you, Sten. Your next question, for sigma metrics calculation, do you recommend to derive the bias from the difference between the EQA assigned value and the lab's value? If not, what would you suggest?
That's a great question. There are indeed multiple ways to calculate the bias, and probably the most formally correct way to calculate the bias is to say, I have an EQA or proficiency testing program. I measure my values, and then I'm compared against peer group values or all group values, or if we're lucky, we have an accuracy-based survey, and we'll get compared to a reference method or a reference material. The downside of calculating bias that way is it takes a long time, and we don't get many data points. So typically in the U.S., 2 to 3 times a year, we're getting maybe 5 samples. So we might only get 15 measurements in a year where we can compare ourselves to a target value or a group consensus value or a reference value, and then determine our bias. A peer group program, on the other hand, well, we can get data off that probably every day. And it will include all of our QC data points, not just a handful or a dozen survey points. So oftentimes in pragmatic terms and ease of accessibility, using a peer group and comparing your mean against the peer group mean is a practical way to determine your bias. A final way to determine your bias is to use an assay control. And if the assay control on the package insert, it says, this, for your instrument, we've tested this and it looks like this should be your mean. And then you look and you calculate your own mean. What's that difference? Well, that difference is also a bias. So my own preference, and again, this is my bias about bias, is to use a peer group program, simply because there's so much data and it's so easily available through most of the peer group programs. So you can update your bias estimates more than once or twice or 3 times a year.
Thank you, Sten. All right, your next question. How often do we need to evaluate an assay to check whether the sigma metric is maintained?
That is a very great question, and I think it's asked very frequently, and I think people worry that a sigma metric is something they're going to have to calculate every day, and then that adds a whole new set of numbers that they have to manage. The sigma metric in practice, in the laboratories that we have in our verification program, we encourage them to do it at least twice a year, but we often find the labs will do it every month. So at the same time they're doing a monthly QC review, they're also taking a look at their sigma metric, because essentially the same data is used. You have an updated CV, maybe you have an updated bias from your peer group. Well, that can help you recalculate your sigma metric. But just because we're looking at that metric every month or every quarter, that does not mean the sigma metric is going to change significantly. In some labs, we've been working with them for over a decade, and their sigma metric fluctuated a little bit, but they didn't change their rules for 9 years. So they had an assay, and it stayed in the same sigma category for the whole lifespan of an instrument. So we don't see a lot of changes in QC, even if we're monitoring that sigma metric on a monthly basis. At least with a lot of the major instruments and the most robust methods, that sigma metric doesn't change significantly very often. So you might check your sigma metric every month, but you're probably going to have the same set of rules for that entire year.
Okay, your next question looks like a three-part question. So this user says, excellent talk. Can you please comment on when QC is commonly run, for instance, immediately after daily maintenance or once a shift? Can different concentrations be run at different times instead of together at the same time?
Mm-hmm. So it might seem like a three-part question, but it's actually an all of the above answer. That very often after you've done your startup routine on the instrument, it's a convenient time to run the QC to make sure everything is working correctly, because of course, wouldn't we all like to know before we start running patients if the instrument is out of control? Then for some labs, and sometimes it's mandated by regulations, or sometimes it's just driven by the professional ethic, that yes, I can run QC once a day and I will do something up front, but I might also want to space and stagger QC throughout the day so that I'm looking at more than just a snapshot of this morning. I'm getting a glimpse of QC in the afternoon, and if we have a third shift, we're getting a glimpse in the night. And some people will indeed take different levels, and so they're running one level in the afternoon, and they run a different level in the evening. So all of those things fall within your professional judgment, particularly in the US, and if you go by ISO 15189, the general consensus is you must at least run QC once a day, and usually it's 2 controls or more. As I mentioned in the presentation, depending on the sigma metric, you may actually need to run more QC, that is, take more measurements, and even run more often, the lower your sigma goes. So there's a regulatory floor under which you're not supposed to go, but then if you know how good or bad your method is, that should also dictate how often you're going to run your QC.
All right, thank you. Okay, we have time for a couple more questions here. Let's move on to this one. I understand the use of sigma for establishing and running QC. Is there an application for anticipating or determining sigma as part of new method evaluation? And if so, how might this be approached?
That is a very good observation, and not only can you use it to determine how you operate, you can use it to determine whether or not you accept the method. So if you think about method verification or method evaluation, that's when you're getting a new test or a new instrument into your lab. Of course, you would like to make this sigma metric judgment to be able to say, this box is good, let's get it into routine use, or to be able to say, well, there's a couple assays on here that aren't very good, and either the manufacturer needs to check and maybe make some improvements or fix something to improve those assays, or this might be considered a box that we don't accept. Maybe it's, you know, the lemon box, and we have to get a replacement instead. So the same data that you use, that you calculate in your method evaluation process, you run a precision study, you do a comparison of method study. Those help you determine CV and bias. So you get the same ingredients for a sigma metric that you do from your routine QC and your proficiency testing. So not only can you use that method evaluation data to accept or reject the method, you can even project out and say, not only do I know if I have a good or bad method, but if I choose to keep that method and put it into routine use, I also have an idea of how I'm going to run QC for that method.
Okay. Now, if an assay is determined as a Six Sigma assay, how many times do controls need to be run?
So the Westgard Sigma rules, when you have a Six Sigma assay, they give you a very simple rule of thumb, which is widen your limits out to 3SD, and you're going to run that. You probably run 2 levels of control if it's a chemistry control, 3 levels of control if it's a hematology control, or, you know, depending on the type of assay, you'll have 2 measurements or 3 measurements, but you won't need any other rules. And the information on frequency tells us when we calculate the acceptable QC frequency rate for a Six Sigma assay, it's roughly about 1 QC measurement, or I should say, you make the QC measurements, whether it's 2 or 3 controls, about every 1,000 patient samples. So for many labs, that just means you have mathematically justified doing QC once a day. But the science now supports what has been up until now an arbitrary practice.
All right, thank you, Sten. We will go ahead and wrap up with this last question. Do you recommend only CLIA or Rillibock for the total allowable error?
That's a great question. It's when we talk about harmonization and standardization, unfortunately, the goals that we use around the world need a standardization and a harmonization. It's a crazy thing, but the goals being applied in Germany by the Rillibock rules are different than the goals being applied in the US and in CAP laboratories by the CLIA goals. Sometimes those differences are quite dramatic. So one of the things we do at Westgard is we try and keep track of all the different goals available across the globe, and we put them in a comparison that you can look at for free. Again, it's in our quality requirements section on westgard.com. And then we ourselves, in our verification program, we make a recommendation. When we choose a goal that we think is optimal, we don't choose the smallest goal because we don't like to choose goals if actually no one on the market can hit it. And there are a set of goals out there where, yes, they may be very scientific, but nothing on Earth is able to achieve that level of quality. So that goal might be something that is aspirational for the next set of research and design engineers at our diagnostic providers, but we need a different goal to govern how we run the tests that we have today. So we make an assessment of the practicality of goals, and in our program we don't try and choose easy goals. We always try and choose a goal that is achievable, but it is also challenging. So we want to make sure that we don't have a goal that, oh, any method can hit this. It's not so much a test as a rubber stamp. No, we always try and assess the performance of instruments and labs out there to figure out how can we challenge them to achieve a little bit better quality in the future.
Thank you very much, Sten. Do you have any final comments for our audience before we go today?
Well, I just want to thank everybody for taking the time. That's the most valuable resource that you have right now is your attention and your time. And the fact that you have been willing to focus on quality speaks highly of your professional commitment. It's really important as you move forward to keep that quality focus in the center of your vision. And I think, I should say, I hope that the tools that we have provided to you help you sharpen your focus on quality and make it easier for you to see how you can get the best methods in your laboratory, how you can get the best operation of QC in your laboratory, and deliver the best results to your patients. Thanks again for your time. I hope you have a good rest of your day.
Thank you again, Sten Westgard, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Abbott, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.